In this podcast episode, Dr. Marc Breedlove, a renowned professor of neuroscience, discusses the complex interplay between hormones, brain development, and sexual orientation. The conversation explores how prenatal hormone exposure, particularly testosterone, influences not only physical traits such as finger length ratios, but also sexual preferences and behaviors in humans and animals. They delve into scientific findings on brain structures related to sexuality, the impact of maternal immune responses, and behavioral plasticity shaped by biology and environment. The episode also touches on unique animal models like gay rams and outlines the challenges in studying sexual orientation through a biological lens. Dr. Breedlove begins by highlighting one of the most consistent findings in human sexuality research: the correlation between the number of older brothers a male has and his probability of identifying as gay. Statistically, the odds of a firstborn son being gay are about 2%, increasing by approximately a third with each older brother. This linear increase reaches a point where having around a dozen older brothers gives an almost even chance of a male being gay. Importantly, this effect only applies to biological older brothers from the same mother, rather than stepbrothers or adopted siblings, and is not influenced by social factors or upbringing. This phenomenon is explained via the maternal immune hypothesis, whereby a mother's immune system reacts to male-specific antigens, particularly a protein called neurologen 4Y, during successive male pregnancies. The antibodies generated may influence the sexual differentiation of the brain in later-born sons, increasing the likelihood of homosexuality. The maternal body's biological memory of past male pregnancies, therefore, might influence the neurodevelopment of subsequent male offspring distinct from environmental or cultural inputs. The role of prenatal testosterone emerges as a central theme in sexual orientation studies. Dr. Breedlove recounts the early skepticism in the scientific community about hormones shaping human sexuality, but discusses pivotal research that challenged this. One notable biomarker is the ratio between the second index and fourth ring digits, known as the 2D/4D ratio. This ratio tends to differ between sexes, with males generally having a lower ratio due to more prenatal testosterone exposure, suggesting a masculinized pattern. Breedlove's own research, conducted in the late 1990s and early 2000s, confirmed that lesbians tend to have more masculinized digit ratios compared to heterosexual women, supporting the hypothesis that higher prenatal testosterone exposure increases attraction to women. However, gay and straight men show no significant difference in 2D/4D ratios, indicating that while prenatal testosterone influences sexual orientation in females, male sexual orientation may depend on how their brains respond to these hormonal cues rather than the hormone levels themselves. The discussion extends to neurological correlates of sexual orientation. A landmark study by Simon LeVay discovered differences in the size of a hypothalamic nucleus, the interstitial nucleus of the anterior hypothalamus 3, INA3, between gay and straight men. Gay men had sizes comparable to women, suggesting neuroanatomical influences on sexual orientation. Although controversy surrounded this research, including concerns around the impact of AIDS on brain tissue and accusations of bias, it has been independently replicated, lending credibility to the findings. This neuroanatomical evidence supports the notion that sexual orientation has a biological basis. However, whether these brain differences cause sexual orientation or result from it remains unresolved. Importantly, this research addresses limitations of earlier thinking that sexual orientation was solely a social or lifestyle choice. Dr. Breedlove emphasizes the use of animal models to understand the hormonal underpinnings of sexual orientation. His lab's work with rats shows that neonatal castration leads to highly female-typical sexual behaviors in male rats, illustrating how early hormone exposure organizes adult sexual behavior. However, the concept of sexual orientation in animals is not straightforward. For instance, male rats will mount any receptive partner, regardless of sex, so attributing orientation in such species is complex. Intriguingly, some animal species provide better models for orientation. The example of gay rams is discussed, where a minority of male sheep exhibit exclusive homosexual behavior, refusing to mate with females despite their presence. Neurological differences have been identified in these rams' preoptic areas, paralleling human hypothalamic findings. This suggests that, at least in some species, sexual orientation and partner preference have identifiable biological substrates, including aversive pathways to one sex. Hormone exposure does not only shape brain and behavior prenatally, but also during development and adulthood. The human brain grows rapidly after birth, maintaining a fetal-like growth rate up to around 6 years of age, allowing hormones and environmental stimuli to influence brain plasticity over a prolonged period. Testosterone levels influence sexual drive and behaviors throughout life. Studies show that adult hormone fluctuations, such as those occurring due to competition or sexual cues, produce changes in circulating testosterone that modulate behavior dynamically. Moreover, hormone replacement therapies in hypogonadal men demonstrate that testosterone increases libido, energy, and mood, confirming continued hormone sensitivity in the adult brain. Observations from unique adult scenarios, such as anabolic steroid use in bodybuilding communities, hint that synthetic androgens can alter sexual preferences and behavior, suggesting hypothalamic plasticity into adulthood. However, such effects are complex and vary widely among individuals. The conversation addresses the challenges of mapping fixed behavioral circuits, as characterized in animal models, lordosis in females and mounting in males, onto the diversity of human sexual behaviors. Human sexuality is not defined solely by motor patterns, but by partner preference, motivational states, and complex social factors. Unlike rigid patterns in some species, humans display wide variability in sexual behaviors and partner choices, complicating efforts to create direct animal analogs. Dr. Breedlove highlights that human sexual orientation involves both attraction and aversion, with possible neurobiological pathways mediating repulsion from certain partner types alongside sexual desire. This duality could explain sociocultural variability in acceptance and expression of homosexuality. Moreover, sexual fluidity appears more pronounced in women, where same-sex attraction and partner choice can change over time, suggesting that female sexual orientation may be more flexible than male orientation. This is consistent with observations of biological differences, such as the presence or absence of aversive circuits. The podcast discusses intersex conditions such as congenital adrenal hyperplasia, CAH, and androgen insensitivity syndrome, AIS, which demonstrate how variations in prenatal hormone exposure or hormone receptor function influence physical sexual development and sexual orientation. In CAH, XX individuals are exposed to elevated prenatal androgens, often leading to masculinized genitalia and an increased likelihood of same-sex attraction compared to typical females, though most remain heterosexual. Meanwhile, individuals with complete AIS have XY chromosomes, but their bodies cannot respond to testosterone due to defective androgen receptors, resulting in typically female physical characteristics and heterosexual orientation toward men. These conditions underscore the complexity of how genetic, hormonal, and social factors intertwine to shape sexual identity and orientation, demonstrating that biology does not determine orientation with absolute certainty, but rather biases probabilities. While biology plays a significant role in shaping sexual orientation and behavior, Dr. Breedlove acknowledges that environment and culture influence other gender-related behaviors, such as preferences for particular toys or activities. Boys, for example, exhibit more rough-and-tumble play that corresponds with prenatal testosterone levels, but can be influenced by social contexts. The brain's plasticity allows for behavioral modulation across the lifespan, although to varying extents. Cultural expectations and socialization may amplify or suppress biological tendencies, yet the data suggests social factors alone cannot explain sexual orientation patterns, especially in light of biological findings. The podcast also touches on how societal narratives and misinformation, such as the outdated association of theater participation with homosexuality, persist despite scientific evidence, reflecting ongoing cultural struggles to understand and accept diverse sexual orientations. Dr. Breedlove explains that sexual differentiation of the brain is not solely orchestrated by gonadal hormones, but is also influenced by the genetic sex of brain cells themselves, especially in birds. This insight comes from observations in gynandromorphic birds, animals with male genetics on one side and female genetics on the other, who display brain and behavior traits corresponding to each side. He intended to conduct grafting experiments in finch embryos, swapping heads across sexes to disentangle the influence of brain versus body gonadal sex on behavior. But the challenges were significant. Current understanding shows that in some species, local hormone synthesis within the brain plays a crucial role in sexual differentiation independent of circulating gonadal hormones. Dr. Breedlove shares his personal journey from a working-class upbringing in the Ozarks to Yale University and ultimately a distinguished career in neuroscience. His curiosity, drive, and fortunate mentorship enabled him to pursue complex questions about hormones and behavior, with sexual orientation emerging as a focal interest. He reflects on the importance of embracing scientific uncertainty, using data to challenge assumptions, and separating biological facts from political or social narratives. The interview serves not only to educate on cutting-edge science, but also to encourage thoughtful and nuanced conversations about human diversity. Throughout the dialogue, Dr. Breedlove emphasizes that sexual orientation is the product of multiple pathways and interacting biological and environmental factors. The fraternal birth order effect, digit ratios, hypothalamic differences, and hormone sensitivity represent pieces of a multifaceted puzzle. Ongoing research aims to further elucidate mechanisms such as maternal immune modulation, neurodevelopmental hormone signaling, and brain plasticity. Recognizing variability within populations and across development remains essential to advancing understanding and promoting inclusivity. Finally, Dr. Breedlove notes his work on a forthcoming book about the biology of sexual orientation, aiming to synthesize decades of research to demystify this profound aspect of human identity.